Hello, fellow neuroscience enthusiasts. I'm Pega, a current postdoctoral fellow at Research Center of CHU San Justin, and this video is one of the installments of the Neuro Discussion series in which we briefly review the results of a selected paper and discuss its relevance to the ongoing research in the field. The paper we are going to talk about is from Dr. Gabriel Puchalon lab and is entitled Restoring Transient Connectivity During Development Improves Dysfunctions in Fragile X Mice. GABAergic inhibitor neurons, although comprising only about 20% of cortical neurons, play a pivotal role in regulating brain activity due to their remarkable structural and functional diversity. These inhibitory neurons represent a highly specialized and diverse population exerting distinct forms of inhibition based on their unique properties and synaptic connectivity. While all GABAergic inhibitory neurons utilize GABA as a neurotransmitter and share a non-pyramidal morphology with non-spiny or sparsely spiny dendrites, they exhibit significant variability in other characteristics that influence their effect on postsynaptic targets. Different subpopulations of these neurons target distinct parts of the postsynaptic neuron membrane, express unique combination of ionic channels, leading to diverse firing patterns and form synapses with varying temporal dynamics. Furthermore, they differ in their sphere of influence, as evidenced by their laminar and horizontal spread of their axonal arbors. The development of the inhibitory neuron is a tightly regulated process shaped by both genetic factors and neuronal activity. In the figure on the left, you can see and compare the radial migration of the excitatory neurons in the cortex, and on the right you see the tangential migration of the inhibitory neurons. These inhibitory neurons are born in specific embryonic brain regions called the MGE and LGE, medial and lateral ganglionic eminences. After being generated in these areas, they migrate tangentially to the cortex, where they establish precise synaptic connections. As inhibitory neurons integrate into the six-layered neocortex, their incorporation follows the inside-out assembly of cortical layers. Early-born inhibitory neurons occupy deep layers, the infragranular layers, while later-born inhibitory neurons are positioned in the superficial or supragranular layers. Cortical connectivity develops in a similar pattern, starting with deep layer innervations at birth, followed by gradual integration of more superficial topographic units. This process is supported by transient circuits involving early generated subplate neurons, which act as temporary intermediaries between the thalamus and cortical or subcortical regions during the maturation of cortical synapses. By the late prenatal period, the earliest inhibitory neurons are already positioned within infragranular layers and have begun establishing their afferent and efferent connectivity. However, it remains unclear whether these inhibitory neurons immediately integrate into their adult circuits or form transient connections that establish the development of mature synaptic networks. Early generated circuits are critical for the maturation of cortical network activity and the formation of excitation-inhibition balance. This process involves the maturation of specific populations of inhibitory neurons, called parvalbumin (PV) and somatostatin (SST) neurons. They are both derived from medial ganglionic eminences, so they share embryonic origins and transcription factors. Despite these commonalities, each cell class exhibits distinct patterns of synaptic specificity in the mature cortical circuits. In the somatosensory barrel cortex, a large fraction of SST neurons target dendrites and provide feedback inhibition to local pyramidal neural network, refining their responses. By contrast, PV neurons are strongly recruited by ascending sensory thalamic inputs and form feed-forward perisomatic inhibition to the same cell that received direct thalamic excitation from. In adulthood, PV neurons provide feed-forward inhibition in response to sensory thalamic activity, while SST neurons mediate feedback inhibition. While PV neurons have been associated with excitation inhibition impairment observed in neurodevelopmental disorders, SST neurons have recently been suggested to regulate and control neuronal dynamics in the developing cortex. Considerable evidence suggests that the development of the PV neuron innervation extends well into late postnatal stages and is regulated by sensory experience during critical periods of plasticity. However, GABAergic synapses are present in the neocortex before synapses of PV neurons appear to be functional. This indicates that another source of synaptic GABA must be present in the developing cortex, forming transient connections that facilitate the development of mature synaptic networks, as I mentioned in the previous slide. SST neurons represent one source that could provide this innervation. Consistent with this idea, the hippocampal network contains long-range projecting SST cells that are present by postnatal day 5 and are particularly effective in supporting immature network synchronization. These hub SST neurons in the hippocampus precociously exhibit mature structural and functional properties, and they share a common embryonic origin with neocortical SST neurons. This raises the question as to whether cortical SST neurons play a role in the functional maturation of the neocortex. In these papers, it's reported that the SST neuron in the infragulanular layers receive dense but transient innervation from thalamocortical afferents during the first postnatal week. During this period, PV neuron and pyramidal neurons as well, within the same layer, receive weaker thalamocortical inputs, yet are strongly innervated by SST neurons. Further, upon disruption of the early but not late SST neuron network, the synaptic maturation of thalamocortical inputs onto PV neuron is perturbed. This result suggests that infragranular SST neurons exhibit a transient early synaptic connectivity that is essential for establishment of thalamic feed-forward inhibition mediated by PV neurons. So until now, we know that SST neurons receive transient connections from the sensory thalamus, yet the implication of transient connectivity in the establishment of excitation inhibition balance and thus the neurodevelopmental disorder remain unknown. Now, in today's paper, we are going to particularly focus on the etiology of Fragile X syndrome. FX disorder. As I mentioned before, excitation inhibition network imbalance is considered as the core of many neurodevelopmental disorders. This excitation inhibition imbalance has been shown to result in cortical hyperexcitability. Hyperexcitability of sensory upper level cortical circuits leads to hypersensitivity and tactile processing abnormalities in this disorder. As I also mentioned before, PV cells are thought to underlie hypersensitivity associated with this disorder or other neurodevelopmental disorders. However, as I mentioned in the previous slide, these PV neurons mature only after critical periods of development, while this FX is associated hypersensitivity is observed at earlier postnatal stages. In contrast, early activity of SST neurons, driven by sensory thalamocortical inputs, suggests that transient thalamocortical connectivity to these neurons orchestrate cortical maturation, and thus play a critical role in the etiology of neurodevelopmental disorders. This developing program of SSC neurons, therefore, could be an ideal therapeutic target in the context of early intervention. However, transient connectivity to SSC neurons in neurodevelopmental disorders remain unexplored. To explore the role of this transient connectivity to SST neurons in Fragile X syndrome, in this paper we'll specifically see that first, somatosensory thalamocortical inputs to SST neurons, which are normally transient, are persistent in a mouse model of this disorder. Second, chemogenetic manipulation of SST neurons within a critical time window regenerates the transient dynamics of these thalamocortical circuits. This restoration of transient thalamocortical inputs to SST neuron alleviates the Fragile X syndrome sensory impairments observed both within cortical circuits and tactile behaviors. And last, SST neurons involved in transient thalamocortical connectivity are an essential developmental trigger of subsequent circuit maturation and processing of sensory inputs.
The aim of the first experiment is to determine if transient connectivity between SSC neurons and thalamocortical inputs play a role in Fragile X syndrome. As I mentioned, they used FMR1 knockout mice as a model of Fragile X syndrome. These are genetically modified mice that lack the FMR1 gene, which encodes the Fragile X mental retardation protein. Thalamocortical inputs are connections between the thalamus, a brain region that relays sensory signals to the cortex, and the cortex. And they wanted to investigate here how thalamocortical inputs influence SST neurons during development. For this, they focus on SSC neurons located in the primary somatosensory cortex, which is part of the brain responsible for processing sensory information. In A, they performed longitudinal analysis that refers to tracking changes over time. And here, they follow the responses of SSC neurons from postnatal day 5 to postnatal day 40 to understand how the connection between SSC neurons and thalamocortical inputs evolves during development in both control and FMR1 knockout mice. They used GFP, green fluorescent protein, as a marker to identify SSC neurons. GFP is expressed in SSC neurons through an AAV diode GFP injection at postnatal day 0 in a specific mice model SSC3 wild type and SSC3 FMR1 knockout mice. This allows them to selectively visualize and track the activity of SSC neurons. Next, they applied electrical stimulation to the thalamocortical fibers in prepared brain slices from different age groups. You see in B, the study groups are divided into three age windows: early, P5, P6, intermediate, P10, P16, and late, P30, P40. These brain slices that preserve the connection between the thalamus and the cortex allows the researchers to study how thalamocortical inputs interact with SSC neurons. In B, you see an example of thalamocortical slice expressing the AV diode GFB in primary somatosensory cortex SSC neurons at P14. In C, you see an example of SSC neuron labeling within cortical plate of primary somatosensory cortex and a schematic illustration of SSC neuron recording. In D, you see the average excitatory postsynaptic potentials evoked in every layer 5 somatostatin cell, following the thalamocortical electrical stimulation in gray and their population average from control in black and FMR1 knockout mice in blue, showing the transient dynamics versus the persistence of thalamocortical inputs onto somatostatin neurons. In E, the black and blue dots represent the populational amplitude of the excitatory postsynaptic potentials in control and FMR1 knockout mice, which represents the strength of the synaptic response to thalamocortical input. In control mice, the amplitude of the excitatory postsynaptic potentials decreases with age, which suggests a reduction in the strength of the excitatory input to somatostatin neurons as they mature. This could be a normal developmental process where transient connections might weaken over time. However, in the fMR1 knockout mice, the excitatory postsynaptic potential amplitudes are significantly higher at all time points compared to control mice. This suggests that the excitatory synaptic inputs to somatostatin neurons are abnormally strong in Fragile X syndrome, which may contribute to sensory processing deficits seen in the disease. Now in F, you see the changes in the charge of the excitatory postsynaptic currents between control and knockout mice. The charge of the excitatory postsynaptic current reflects the total amount of synaptic current over time and can give insight into the overall activity level of the neuron in response to the thalamocortical input. As you can see, similar to the amplitude, the charge of excitatory postsynaptic currents also decreases with age in control mice. This suggests a developmental reduction in the total synaptic activity that somatostatin neuron receive from thalamocortical inputs, consistent with the idea of transient connectivity that weakens over time. As you can see, similar to the amplitude, the charge of excitatory postsynaptic currents also decreases with age in control mice. This suggests a developmental reduction in the total synaptic activity that somatostatin neuron receive from thalamocortical inputs, consistent with the idea of transient connectivity that weakens over time. In this figure, they investigated whether restoring transient thalamocortical connectivity onto somatostatin neurons can correct somatosensory impairments in fMR1 knockout mice. As it has been shown in the previous figure, thalamocortical connectivity to somatostatin neuron is transient, but it remained persistent in fMR1 knockout mice. Since metabotropic signaling regulates this transient connectivity, the researchers used chemogenetic modulation using DREADS to mimic normal developmental dynamics. In this figure, you see the early rescue strategy using AAV-DIO HM3DQ mCherry, which stands for adeno-associated virus. DIO stands for double flux inverse open reading frame. HM3DQ is a human muscarinic tree designer receptor, and mCherry is a red fluorescent protein. This construct was injected into SST3 fMR1 knockout mice at P0, P1, while controls received AAV-DIO GFP. Afterward, clozapine N-oxide or CNO injections were administered daily from P6 to P16, covering early and intermediate developmental periods where thalamocortical inputs to somatostatin neurons normally mature. This aimed to restore transient thalamocortical input dynamics in fMR1 knockout mice. In the results, they recorded the excitatory postsynaptic currents in somatostatin neurons during the intermediate period from P10 to P16. And they are showing that injecting fMR1 knockout mice with AAV-DIO HM3DQM cherry at bears P0 and activating somatostatin neurons with CNO from P6 to P16 helped to restore normal brain activity. The abnormal excitatory signals in fMR1 knockout mice were reduced to levels similar to control mice. As you can see in panel C and D, the amplitude and the charge of the excitatory postsynaptic currents in the early rescue group were significantly reduced compared to the untreated fMR1 knockout mice, bringing them closer to the control level. In D, you can see that both the amplitude and charge of the excitatory postsynaptic currents evoked in somatostatin neurons within control, fMR1 knockout mice, and rescue conditions plotted as a function of time windows. In E, you see loose cell attached recording of somatostatin neuron firing upon thalamocortical fiber stimulation, and an illustrative example traces of somatostatin cell firing. The inset is showing a high magnification of an action potential. Somatostatin neurons in fMR1 knockout mice fire too much when stimulated, but early rescue prevented this overactivity, as you can see in both panel E and F. This figure demonstrates how early chemogenetic activation of somatostatin neurons restores sensory processing and circuit integration in adult fMR1 knockout mice. The key findings are: first, somatostatin neurons regulate the maturation of layer 4 excitatory neurons and parvalbumin interneurons during early postnatal stages. And second, early intervention improves tactile discrimination by stabilizing neuronal firing patterns. Panel A shows you the experimental timeline where somatostatin neurons are activated using DREADS during P10 to P16 via daily CNO administration, and then the behavioral and circuit analysis occurs between P13 to P15. This timing aligns with a critical window for synaptic connectivity and cortical maturation, aiming to see if early intervention prevents long-term dysfunction. In panel B, you see loose cell attached recording configuration. This panel describes the electrophysiological approach used to study the activity of layer 4 excitatory neurons. The aim of this panel is to determine if fMR1 knockout mice exhibit reduced excitability and whether the rescue condition restores firing precision. Using this technique, electrical stimulation is applied to subventricular zone afferents, projecting onto layer 4 neurons in the same cortical column. Loose cell attached recording allows for minimally invasive monitoring of neuronal firing without altering intracellular conditions. This technique was performed in vivo, not in culture, because investigating the activity of layer 4 neurons within the somatosensory cortex in response to the stimulation of subventricular zone fibers requires intact cortical circuitry, which are preserved in vivo but not in culture. Here is what loose specifically means in this context. The pipette is gently suctioned to make a loose connection, but it does not puncture the cell membrane or form a gigaohm seal. Like this, it preserves the cell's natural intracellular environment. For this reason, this technique is applied to detect the small extracellular voltage changes produced by the neuron's action potential. These spikes are recorded as current or voltage deflections in the pipette. And unlike whole-cell patch clamp recording, which measure intracellular activity, loose cell attached recordings are ideal for simply counting spikes or monitoring firing patterns. The precision in action potential firing is quantified by the inter-spike interval distribution or the variance in spike timing across trials, and a lower variance indicates more precise firing. This precision in firing is shown in panel C. The top trace shows the response of a single trial for a neuron in the control and fMR1 knockout mice. In control neurons, the action potentials are sharp and occur at a consistent time after the stimulation, while in the fMR1 knockout mice, the action potentials are less consistent in timing and often have delayed or missing responses reflecting the imprecision. The bottom raster plot contains 110 trials, providing a visualization of spike timing across multiple trials. Each row corresponds to one trial. In the control neurons, the spikes are tightly clustered in time, forming discrete, consistent bands shortly after the stimulation. This demonstrates high precision in firing across trials. In the fMR1 knockout neurons, the spike timing is more dispersed and variable across trials, with spikes occurring at different times or even failing to fire at some trials. This indicates reduced precision and increased trial-to-trial variability. The fMR1 knockout neuron's lack of precision and variability is quantitatively analyzed and summarized in panel D and E. The panel D is showing the cumulative probability of trials without action potentials across three groups: the control, fMR1 knockout mice, and the rescue group. X-axis is showing the number of trials without action potentials, and the y-axis is showing the cumulative probability from 0 to 1. The key finding here is that fMR1 knockout mice show a higher probability of trials without action potentials, indicating reduced neuronal firing. The rescue group's curve shifts closer to the control, suggesting the rescue strategy partially restores normal firing probability. Please note that if fMR1 knockout mice had a higher probability of trials without action potentials, you would indeed expect their line to reach the top, the cumulative probability of 1, sooner, because they would accumulate trials without action potentials faster. However, the plot shows the opposite. They reach the top slower, indicating they have fewer trials without action potentials before reaching the same probability level as control. This reflects a different aspect of reduced firing. They might not be completely silent, but they fire less often or less reliably, leading to a slower accumulation of trials without action potentials in the cumulative plot. Panel E provides neuronal activity post-stimulation. The heat map provides a summarized view of the firing intensity for each neuron over a 35 millisecond window. The 35 millisecond is divided into 1 millisecond bins, resulting in 35 bins. Each row in the heat map represents one neuron being monitored for 35 milliseconds after a stimulation being presented. We call that one trial. For each neuron, the number of action potentials in each 1 millisecond bin during the single trial is quantified. The count of action potentials in each bin is represented by color intensity, with darker colors indicating higher firing. In comparison with the raster plot in panel C, which gives us a detailed view of when each neuron fires, showing the precise timing of action potentials, the heat map provides a visual summary of how often each neuron fires over time, allowing for a clear comparison across different experimental conditions: the control, fMR1 knockout, and the rescue condition. Panel F displays bar graphs comparing the mean firing probability of neurons across 300 trials. X-axis represents the experimental groups, and y-axis shows the mean firing probability ranging from 0 to 1, indicating the probability of neurons firing in response to the stimuli. The control group typically shows a higher mean firing probability, indicating a robust neuronal response to the stimulation. The fMR1 knockout group exhibits a significantly lower mean firing probability, reflecting the impact of fMR1 knockout on reducing neuronal excitability. And in rescue group, the mean firing probability is increased in comparison to fMR1 knockout group, moving closer to control levels. That suggests that the rescue strategy partially restores the normal firing pattern. This panel shows three correlation matrices for different experimental groups: the control, fMR1 knockout, and the rescue. Both X and Y axes represent individual neurons, with each cell in the matrix indicating the correlation of activity between pairs of neurons. Darker squares indicate higher correlation, while lighter squares indicate lower or negative correlation. Here is how the matrix is being generated. For all the trials, the activity of each neuron is recorded post-stimulation over a 35 millisecond window. Each neuron's spiking activity is binned into 1 millisecond intervals, resulting in a 35 bin time series per trial for each neuron. Then, for each neuron, the average number of times it fires in each 1 millisecond bin across all trials is calculated, creating a vector of mean firing rates over time. This vector represents the average activity pattern of the neuron during the stimulation response period. The covariance between two neurons' firing rates over time is computed using their mean firing rate vectors and is presented as one element in the correlation matrix. And the Pearson correlation coefficient is a normalized version of the covariance that standardizes the values to range between -1 and 1. A positive correlation means the neurons tend to fire together. A negative correlation means one neuron tends to fire when the other is silent, and a correlation of 0 indicates no consistent relationship in their activity pattern. And here is how this correlation matrix provides insight into functional connectivity between neurons in response to the stimulation. For the control group, the matrix shows a mean correlation of 0.1, suggesting a moderate level of coordinated activity among neurons. And for the fMR1 knockout matrix with a mean correlation of 0.2, there is an increase in the correlation compared to the control, indicating a change in the pattern of neuronal coordination, potentially due to the knockout affecting normal network dynamics. For the rescue matrix, the mean correlation is closer to control than fMR1 knockout, suggesting that the rescue strategy partially restores the normal pattern of neuronal coordination. Panel H is representing the statistical distribution of neuronal correlation. In this panel, the x-axis represents the mean Pearson correlation values, while the y-axis, labeled as iteration number, represents the number of times a certain mean Pearson correlation value was obtained. The use of iteration as y-label suggests that the data shown is likely from a bootstrapping or permutation analysis, where the mean Pearson correlation is calculated multiple times or iteration using different reassembled datasets. The distribution of these values is plotted as the smooth curve in black, green, and red. The green and red distributions show comparisons involving the fMR1 knockout group, and the blue dashed line indicates the actual mean Pearson correlation observed for the FMR1 knockout group, which lies at the higher end of the distributions. The statistical significance suggests that the FMR1 knockout group shows significantly higher mean correlations compared to the control plus rescue group, but there is no significant difference between the rescue plus FMR1 knockout and control plus FMR1 knockout groups. Panel I shows a histological section of the brain in activated and neighboring columns with CFAS staining, a marker of neuronal activity in response to whisker stimulation. The aim is to assess how genetic manipulation affects neuronal activation patterns in response to sensory input, comparing control, FMR1 knockout, and rescue groups. As you can see, FMR1 knockout mice shows significantly higher CFAS intensity compared to controls, suggesting altered neuronal activation in layer 5B and layer 2-3. The rescue group shows a partial normalization toward control level, though not fully significant. Similar analyses are performed in a neighboring non-stimulated column, and you can see similar trends as in the activated column, with FMR1 knockout mice showing increased activation and rescue group showing some corrections. Now about panel G. The aim here is to evaluate if there are changes in the inhibitory interneuron population due to FMR1 knockout, and whether the rescue strategy can restore this. The panel shows a fluorescent image of PV-positive neurons in cortical layers, and the bar graph is comparing the density of PV neurons per square millimeter across control, FMR1 knockout, and rescue groups. Control and rescue groups show similar PV neuron densities, while FMR1 knockout has a slightly lower density, though the difference is not significant. The aim of panel L is to investigate the interaction between PV inhibitory neurons and CFAS in response to stimulation. The panels show merged images of CFAS in green and PV in red in brain slices from control, FMR1 knockout, and rescue mice. The graph quantifies the percentage of PV neurons that are also CFAS positive, and it shows that FMR1 knockout mice has a significantly higher percentage of colocalization compared to control, indicating more PV neurons are activated. And the rescue significantly reduces this back toward the control level. And the aim of the last panel is to assess the behavioral impact of FMR1 knockout on texture discrimination and the effectiveness of the rescue strategy in restoring this cognitive function. Panel L is showing the novel texture discrimination assay paradigm, in which, in day 1, the mice are being habituated for 10 minutes. In day 2, the mice are habituated, followed by learning and testing phases over several days, where mice are exposed to familiar and novel textures. The graph shows normalized time spent by each group exploring familiar versus novel textures over 10 minutes. Control mice show a preference for novel texture, spending more time exploring them. FMR1 knockout mice show no significant preference, indicating a deficit in texture discrimination. And the rescue mice regain some ability to discriminate, showing a significant preference for novel textures similar to the control group.